We're going live. Ladies and gentlemen, thank you for coming to our show here on The Latest With. Last time we had Dr. Warren Sherman from Columbia University, and this time we are honored to have Dr. Bob Harman, who is the CEO of MediStem. MediStem has always been a very exciting company to us because it's the first company literally to commercialize stem cell therapy, in my mind, having treated thousands of canine and equine patients in the beginning. And basically, as my background being immunology and being used to playing with mice, it's very refreshing to see that we have access now to large animal data from collaborations with people such as Dr. Harman from MediStem. So with that said, Bob, maybe you can begin by telling us a little bit about your history and how you started the company MediStem.
Sure, Tom. Thanks a lot for inviting me, by the way. It's exciting to be able to reach out and get this information to people who are interested in stem cells, and you have a big following. So I'm a veterinarian by training, and I was involved in preclinical and clinical trials for veterinary and human products for about 15 years. In 2002, I had sold those companies and retired from practice, and I was contacted by folks in the stem cell industry who had been working with stem cells from adipose tissue. And they asked, I was a veterinarian, would there be interest in licensing? And we spent about a year looking at the data and ended up licensing this technology worldwide to use stem cells from fat tissue. And I know, Tom, you've talked about a lot of cell types on your show, and you're involved in menstrual stem cells and bone marrow stem cells and adipose stem cells. So there are a lot of sources. VetStem started in 2002, primarily using stem cells from fat tissue in an autologous manner or from the same patient, and extracting them and using them in their fresh state, so the stromal vascular fraction or the fresh mix non-cultured cells. And now we're 8 years later, we're over 3,000 dogs and over 4,000 horses clinically, where we've used them mostly in orthopedic diseases. So that's been the bulk of our experiences in orthopedics, although, as you know, Tom, we've also worked a little bit in immunologic diseases, immune-mediated arthritis, renal disease, also in liver disease. So that's just a really brief sort of how we started. And we're based here in San Diego, and the veterinarians we work with are all over North America, Canada, US, and we've got affiliate licensees in Dubai and in Australia now, and they're actually using allo cells, which we might talk about later as well.
Excellent. Well, Bob, before we jump in, we actually got our first question from somebody named Brandon. Dr. Harman, what is the difference between a mouse mesenchymal, a human mesenchymal, and a dog mesenchymal stem cells? Are the stem cells? That was a question. What do you think? Are there differences?
Yeah, but it's a really good question, because we often use data from another species. So like if you were doing a clinical trial, right, Tom, you'd like to use data from the dog. When we're doing a horse, we'd like to use data from mice. In my view, sort of as a cell-based scientist and a veterinarian, if you take stem cells from various tissues and from various species, but they're large animals, and you're testing them or evaluating them in real diseases in those large animals, and a large animal may be anything from a rabbit on up, the diseases in the cells appear to be very similar. In mice, I always have concerns because they're inbred animals, they're bred for particular uses, and both their cells and the models that we use in mice are interesting and give us interesting ideas about how these cells might work, but they're not quite the same. So although mesenchymal stem cell from a mouse might in the lab in a petri dish look very similar to a mesenchymal cell out of one of the dogs that we treat, I think the diseases and maybe the function of those cells might appear to be different. But when you look at human, and you look at dog, and you look at horse, their cells, I think, from the literature I read, look like they function in a very similar manner. And that's why we now publish with people like you, Tom, and others in the human field, and our other colleagues in veterinary, because I think the cells are pretty similar.
Oh, okay. We won't spend all of it on questions, but two more questions came up. So let's just quickly address it so the people continue
Sure.
watching and telling their friends. Um, okay, so, wow, Carl, does your technology expand MSCs when treating dogs and horses, or do you just isolate and re-inject?
Exceptionally good question, because you always have to ask the question, where do the cells come from and what have you done to them before you can evaluate the kind of data we're going to talk about today. And we do both. We actually use cells directly isolated from fat tissue, and you use an enzyme and centrifugation and washing to get that mix of cells, which is partially adipose derived stem cells, maybe a third of the cells. There are a lot of endothelial progenitor cells and other adult cells in there. So that mix we use in dogs could only be used in the same patient because there's a lot of adult cells. We also at VetStem, when we run out of those cells from the first collection of the first surgery, we will then put those cells into cell culture and expand them. So now we have a little more homogeneous population, and we can expand and make a lifetime supply for a particular dog. Again, we use that because of FDA regulations only for that animal. But at that point, those are the same as what you might use, for example, what Osiris uses in treating with bone marrow expanded mesenchymal cells. And then they can be used, truly as a donor cell to go into a different patient or recipient.
This is a very interesting line of thought, and we will go into the presentation in a little bit, but really quickly, Bob, in my own mind, the question, companies like Astrum are doing 12-day cultures of autologous human bone marrow, and one of their points, it's a great company, as you know, they're almost in the phase 3. They have the accelerated approval for critical ischemia, space in which MediStem obviously is very interested in, because that's where we're doing our clinical efforts. Do you believe that this culture in vitro expansion can also select out the young cells, that it's a good way of quote unquote regenerating, having a young population in which a percentage of more potent and younger cells are in there? Do you think that may happen when you do this culture?
Oh, be careful, Tom. This is live TV. You don't want to ask a question. You don't want to hear some really odd opinion. But, sort of joking aside, the cells change when you put them in culture. The mix changes clearly, and everybody knows that. The adult cells generally don't stick to plastic, and so you tend to select for a more homogeneous population of, quote, stem cells. But also, I think those particular cells on a cell by cell basis, I think start to lose potency as you grow them. Here's why I think that. So if you look at when we treat a dog with a knee arthritis, so, a bad knee, arthritis in the knee, we use between 3 and 4 million nucleated cells. So that's the mixed population. As you know, there's anywhere from 5 to 30% quote true stem cells in there, but there's a lot of other helper cells, including the T regulatory cells, including other adult cells, including other immune system cells. And we get about 75% of all the dogs treated with that 3 to 4 million total cell mix have a fairly profound clinical improvement. Okay, and we'll look at some of that data later. Now, tell me if you remember how many cells in the phase 2 study that Osiris is doing with a donor highly expanded cell population, how many they put into the knee of a human? You remember?
Was it 20 million? I forgot.
400 million.
400 million.
Now, Dr. Kaplan, I'm one of his disciples, and I believe a lot of what he says, he believes they're overdosing, that's just where they started and they thought they needed that many cells. But we see the same data when we use an adipose expanded cell and a vascular stromal fraction unexpanded cell, we need 2 or 3 times as many total cells with the expanded population to get the same clinical effect. So, and it makes sense. You're putting those cells in a non-natural environment. They're sticking to plastic. They're spreading out. They're taking in nutrients from an artificial media, although it's supplemented maybe with some serum. But I think those cells lose a little bit of their potency, so you need to put more of them in.
I see.
But one of the things you get, Tom, as you know, you get a more pharmaceutic-like population, a repeatable population of cells, which is very, very important if you're trying to create an FDA product, that you can have the same number of cells with the same outcomes. So I think, personally, I believe you make up for it by putting more cells in, but I think that probably answers the question. Don't
Yeah,
want to
no, that answered the
run on a
question.
little bit.
That answered the question very well.
Yeah.
Like, really quickly, because it's, one of the things we love about this TV show is that we have experts like you, and we can pick their brains and have really nice discussions. If you take a mouse and you do serial transplantation of hematopoietic cells, does it run out? Do the cells end up dying? Do you remember the answer to that? Because I forgot. Like, is telomerase keeping a stem cell to forever be a stem cell, a bone marrow stem cell? Or is it like in the in vitro system, do they eventually exhaust themselves?
They do, and there's some really interesting data that was presented at the first veterinary regenerative medicine meeting, which, yes, there is such a thing, and it was last March in San Ynez, California, and there were about 250 veterinarians from all over the world that came to talk about veterinary regenerative medicine, and Martin Vidal, who is a D.V.M. PhD from UC Davis, from the vet school, presented data on cord mesenchymal cells, not cord blood, cord mesenchymal cells, adipose mesenchymal cells, and bone marrow mesenchymal cells. And he looked at the number of doublings and how fast you get senescence or dying or differentiation, terminal differentiation in culture. And the bone marrow senesced or died or lost its telomerase ability to be multipotent twice as fast as the adipose stem cell or the cord stem cell. Cord and adipose were quite close. So as you grow them, the more doublings, the faster they lose their ability to be stem-like and ability to be multipotent. And so it's not an unending ability to continue to multiply these cells.
Mm-hmm.
So we actually have a passage 4 limit where we don't go beyond that, even with cultured cells and trying to treat animals, because we think you start to lose, for example, you lose the ability to adhere to a tissue and migrate. I think that's one of the early things you lose. You lose ability to differentiate into some of the cell types.
Yeah.
So
Yeah. No,
at least that's what I believe.
very interesting. We could talk for hours about this. There's a couple of questions here.
Yeah.
What other ones? What relative advantages of adipose versus bone marrow? You touched upon that. How many more mesenchymal stem cells are there in fat as opposed to bone marrow? That's an interesting question.
Yeah, it's a really interesting question, and there was a lot of sort of misinformation in the literature in the early days, I think, about that, because they were looking only at culture adherent populations. But if you look at the cell surface markers that tell you you've got a mesenchymal stem cell-like population, about a third of all of the nucleated cells in an adipose extract are a true mesenchymal stem cell, and
You
that was
said
done
Bob, 1/3 you said?
1/3, and that's data from Yoshimura in Japan
Mm-hmm.
and Colas in the Netherlands
Yeah.
and Varma in Italy, all 3 of them, and they did that with flow cytometry.
Yeah.
And so there's a whole population of cells that don't stick to plastic.
Uh-huh.
And so our old methodology of just saying, well, if it's stuck to plastic, what percent of the cells would tell you how many were in there? And I think that's probably not true anymore. We don't believe that to be true. But if you look at bone marrow, fresh bone marrow extract, fresh adipose extract, looking at the mononuclear fractions in there, got about 1,000 times more stem-like cells, progenitor cells in the adipose tissue as in bone marrow. So
1,000-fold.
yeah, so the starting population's very, very rich in adipose, and we looked at both cell types and trying to decide what to use, because we actually had access to IP on both of those sides. And adipose was so much easier to both collect the fat, process it, and so picture a bone marrow extract is what's the average bone marrow extract in a human? Maybe 25 to 50 mils.
Yeah.
And even in a dog, we can get a kilogram or 1,000 mLs of fat in a live harvest. You don't have to, you could just actually harvest that much. As anybody who has a chubby dog knows, you can get a lot of fat. So you've got not only now maybe 10 to 100 times more weight of fat you start with, and you've got 1,000 times more cells. So the starting material is way more concentrated in terms of the material that you then to start multiplying with. So now, and the reason that's important in a commercial sense is the number of doublings you need to do to get to 10,000 or 100,000 commercial doses of stem cells when you're making a pharmaceutical grade product is so much shorter. So now the cells are younger, fresher. They don't have near as many doublings. They haven't lost as much of their ability. So I believe.
Yeah, this is what those guys in Spain, what is the company, Celerix or something? They're
Yeah,
doing.
yes. Yeah, Solarix is doing exactly that.
Interesting. Okay, there's a couple of
Yeah.
questions, but we apologize to the nice people who ask questions because I want to get into the presentation. So Bob, take it away.
Good. So we'll try to quickly go through, and please stop me, obviously, anytime, Tom, if you
Okay.
want to go down the rabbit hole and talk a little more about some particular data. But I put together a few slides that we use in talking with veterinarians about the evidence for stem cell therapy. And everybody hears about evidence-based medicine. And, you know, here, this poor veterinarian in the middle of this picture has all of these outside influences. They need to worry about safety. How severe is the disease? What are the alternate choices of therapies? Is there any efficacy data, you know, that actually shows, yes, the cell therapy works? What do the owners want to do? What are the relative costs? The ability of the animal even to tolerate certain kinds of drugs like a Vioxx-like drug. So there's this whole pyramid of evidence from in vitro bench research through laboratory animal studies, editorials and opinions of guys like Jeff Gimbel and Arnie Kaplan and Emmett Patel and other people that write papers. And then you have case reports and case series reports, retrospective case control data, and then the prospective uncontrolled and what's considered sometimes to be the gold standard, a controlled study, so a blinded placebo-controlled study. And I'll touch just briefly on each of these kinds of data that lead us to believe as clinicians and as owners of animals and as colleagues that yes, cell therapy in fact has enough evidence that we should be using it in clinical cases. So that sound reasonable, Tom? Great. So and this is looking only at joints. So there's this whole set of data on any of these topics for sure. And so if we're looking at joint therapy mechanisms, chondrogenesis, or the production of cartilage, is the subject of hundreds of papers. Lubricant production, which is the subject of making lubricating molecules in a joint, and stem cells, in fact, do make that. And this was a paper, and we could spend a whole hour talking about this paper where the folks at Sac Med Center actually went through and showed that a stem cell from the fat pad of the knee, mesenchymal progenitor cell, can make this lubricating molecule. Quite exciting. And then Darwin Prokoff, who's one of the giants in our industry at Tulane, now at Texas A&M, showed that these cells produce a blocking protein that blocks interleukin-1. Interleukin-1 is one of the nastiest cytokines, causing a lot of the damage inside of joints. So now let's look at a rabbit. So remember the question earlier, a rabbit is not a dog, is not a human, is not a horse, but it's a place to start where we can actually look at this data. And in this particular study, you can actually look, and I'll see if I can use my pointer, and hopefully that works for us here. If you look at the two groups, so the adipose-derived stem cell group actually had adipose-derived stem cells with a fibrin glue that were placed into a full thickness through cartilage all the way down to bone lesion in the femur of a rabbit. So it's an area where there's cartilage and would be wear and tear. And then in the control group, blinded, there was just fibrin. So a glue, fibrin glue, which comes out of serum, was placed. And if we look all the way over here at 24 weeks, you can see quite easily that the thickness of the cartilage there in the top where the two red arrows are pointing is much thicker in the stem cell group than in the fibrin group. So it showed that you could make, actually, in looking at this if you read this paper by Nathan out of Singapore, it's actually new hyaline cartilage. So any of you that have missing cartilage in your knee, this was evidence that yes, maybe we could actually repair cartilage in a knee.
But this is with fibrin glue.
Yeah, it was fibrin glue in the bottom, which is the control, but in the top, that's adipose-derived stem cells mixed with fibrin glue. So that's actually put in a hole in a lesion. So that's not just injected in a joint, right? And
Yeah.
so rabbits aren't people. So now let's look at, and you know this paper well, Tom, you've seen it before. This is the paper by Murphy and his group, and the study was done at Tufts University at the vet school. And this was Osiris's original study done looking at goat arthritis. So the way this study was done, again, they did a marrow harvest and isolated cells from bone marrow and grew them up. Then they actually took and cut the anterior cruciate ligament and did a medial meniscectomy, meaning the whole meniscus, medial meniscus, was removed. And then they waited 2 weeks. Then they exercised these guys. Okay. So these animals are now exercised, running them up and down and jumping over some jumps. And so you can imagine with no ACL and no medial meniscus, they got severe arthritis. So that was about a month. Then they injected either mesenchymal stem cells or just a dilute vehicle. Okay, so this is not putting it on a lesion. This is actually just injecting these cells or vehicle, the blinded saline control group, just into the joint, into the knee joint. Okay, then they waited 6 weeks or 20 weeks and monitored them clinically. So the veterinarian, Carl Kraus, who's a friend, said it was obvious that it was a blinded study. It was obvious that there were a group that were better and a group that were worse. And in fact, the outcome was as such. So the treated group with cells had a dramatically reduced clinical lameness score. Hey, that's not too surprising based on what we know in the dog and the horse. What was surprising was that they regrew the meniscus in 7 of 9 of the treated group and none of the controls. So if we had a group right now, Tom, of 100 people or 1,000 people watching this show, there's probably 5 or 10% of them that have bad meniscal damage in their own knee. And so this now has led to, for Osiris, to actually do a human, they're in phase 2 clinical studies now, injecting their bone marrow-derived mesenchymal cell into the knee. And so this led us to believe maybe we should consider in the horse and the dog that we could impact the course of clinical arthritis.
Before I forget, Bob, I always forget this. What is Osiris's osteocell product?
Yeah. Oh, so that's an odd matrix material that is not really a stem cell product per se, and I'm sure there are growth factors in there, and there's some, but it's a decellularized like product, I believe, that they're using for bone regeneration.
Okay.
So it's bone specific. It's not a cartilage product.
And that was on the market, right?
It is. It's already on the market. Their chondrogen product, which is this one that they're going after, stem cell in the joint for osteoarthritis, I think is in phase 2 now. It might be moving to phase 3.
Okay.
So if you look at the canine elbow joint, which is very similar to a human, except they're weight bearing on that, obviously, you can actually grow. So that bottom picture is actually a picture of the inside of an elbow joint, and all this dark brown is pure bone. So the cartilage is totally missing. And I don't know if I can get the video to run on this. I'll maybe run the video at the end if we have time. On the right was after just stem cell, you get nice new clean cartilage formation. And it's not in every joint you get that, but you can actually grow new cartilage. And this is not in an experiment. This is a clinical case. This dog actually was very badly lame, treated with stem cells, and at this point they looked in with the scope. This dog was back doing agility, which means they go up and down ladders and run through jumps, and it's a very, very, very rigorous sort of sport that they do. This is another sport dog, and it's actually a working dog. So if you can see that German Shepherd, that's a police dog in training and competition. They're Schutzen dogs. And so anybody who's ever had a bad muscle tear, in the dog, actually in the back end, right where that red pointer. Can you see the red pointer, Tom? I want to make sure you can see it.
I can see it.
Yeah, okay, so on the hind leg, it's like sort of like the hamstring muscle. It's called the semitendinosus muscle. Tears when they make these big jumps and grab hold of the bad guy's arm and pull, and that's their competitions because they're police dogs. That's what they do for a living. This veterinarian, Dr. Gary Brown, in Northern California, has been treating these kind of dogs for 30 years. He just presented this data about 2 weeks ago at the AVOR meeting here in San Diego of orthopedic surgeons, veterinary orthopedic surgeons. And he said in his 30 years, he's never had any kind of therapy, surgical or otherwise, that could repair these bad muscle tears. They all get fibrous tissue and end up retiring early. And he treated 5 consecutive dogs with stem cells injected into the muscle tear. Okay? 5 out of 5 back to full work. And the next picture will sort of tell the story. So if you look in the top in the little graph, it's the name of the dog, the number of cells injected. So this is total cells, as you remember, Tom, not just stem cells. This is this mix of fresh extracted adipose regenerative cells. The pre-grade score, so 4 is very severely affected, meaning they probably can't walk. A 3 would be severe, a 2 would be moderate. So these are moderate to severe cases. And at anywhere from 10 to 19 weeks after treatment, they were graded a 0, meaning they're back to work. Of these 5 dogs, 2 went on to national championship to actually win championships. Unbelievably striking. This is not a blinded placebo-controlled study, but this is a veterinarian who said in his 30 years as a board-certified veterinary orthopedic surgeon, he has never seen these heal and the healing hold. These dogs are all back to very significant work.
Sure.
And I know you're not used to looking at ultrasounds, but in the bottom you'll see ultrasounds. The big hole right there by the red, it's that big multiloculated hole is the tear in the muscle. And in the right picture is 84 days after treatment, and that hole is filled in and gone back on to heal.
This is very, very impressive. But let me ask, the cells here are injected without any matrix, just injected?
Correct. So this is the extracted cells. It would be about 1/3 stem cells, about 1/3 endothelial type cells, which in this you can imagine very important to bring vasculature in and heal, just like your endothelial type cells that you use. So it brings in new vasculature and reduces, most importantly, it reduces the fibrosis, because anybody who's had a bad muscle tear, if it just heals sort of on its own, you get fibrosis, and that's why you go to a physical therapist to break down that fibrous tissue.
Because we have the paper, I think you remember it, where the stem cells were injected into the boy with Duchenne's muscular dystrophy.
Yep.
And so this is very, very interesting. Who else has done intramuscular stem cell injections like this?
Yeah, very little on intramuscular. Lots of intratendinous and intraligamentous injections. We've done over 4,000 in the horse. And there's a lot of studies done with other kinds of products injecting intramuscular. But I think this manuscript's been submitted. So I think it will be the first clinical set of cases of these kinds of muscle tears. Imagine these in the sports population. We just got done watching the Super Bowl, and baseball players with shoulder tears and lots of fairly severe muscle tears that result in lots of fibrous tissue. So it makes sense that it would work.
No, exactly. If I remember correctly, in some of the papers on some of the descriptions of mesoblast, I believe mesoblast was at one point doing this kind of medicine.
Yeah, and most people, like when you injected for Duchenne's, you were trying to get those cells to make a new product. So having a matrix, and you want them to engraft and stay there, a little different kind of functionality than where we're trying to get them to actually repair a tear in a tissue. So I think it's a slightly different approach, but the exact same concept of getting those cells directly to the injury, as opposed to going by an IV or vascular route. I think with this bad a muscle tear, they would get there. But boy, when you can just stick the cells right where they need to go, it makes sense to do so, to me.
Yeah. And do you have to prepare the muscle in any way before the injection? I mean, I imagine there's a lot of inflammatory stuff going
Yeah.
on there?
That's a really good question, and I would be surprised if you don't have 10 people ask that question if you had a group of clinicians. And the reason is that I think there was a misnomer that people thought, well, boy, you got to wait until an injury sort of calms down before you put the stem cells in there, because maybe they would die off more
Yeah.
because of all of the white cell, all the white cell mediators that get dumped. I think it's the opposite. The mediators are the biggest attractant of stem cells naturally.
Mm-hmm.
And if you look at the list of inflammatory mediators that attract in stem cells, there's a whole host of them, very well published now. I think the cells do very well in there, and the object is to get them to turn off the inflammation before you get fibrosis. So our belief is now in tendons in horses, in ligaments in horses, and in dogs, and in these kind of muscle tears, that the sooner you get the cells in there, the better, in terms of reducing the damage. And as you know, Tom, these cells are also anti-apoptotic, and there's a lot of cell death going on in a stroke, in a muscle tear, in laminitis in a horse. And I think these cells, as opposed to just trying to make new tissue grow, actually block the death of the tissue in there. And, picture anybody in a car accident, whether it's a dog hit by a car or a human with polytrauma, and you've got all of those cells that have been ischemic that are starting to die. It's like the work that you've done with Amit Patel and Dr. Perrin and other people in cardiovascular. It's a whole lot easier to block cell death than it is to try to build new tissue. So I think that's another function of these
Well,
cells.
Bob, along those lines, and
Yeah.
one of the things we're trying to do with this TV show is to really have open network for ideas and idea exchange, and basically try to unify this field. One thing that just came to my mind that you should think about doing is, have you ever thought about sepsis, using these cells for sepsis?
Yeah, so if we got to the end of this publication, this presentation I'm doing that I normally do for veterinarians, I've got a beautiful slide on some of the data that you're well familiar of, looking at sepsis, and GI tract inflammation, which is where a lot of the sepsis comes in. And we actually treat inflammatory bowel disease, which is similar to ulcerative colitis and Crohn's in the human. And I actually have personal experience with a pet of my own. My wife and I own a beautiful white cat that got really bad inflammatory bowel disease. And if you don't block that, they will get sepsis. And we treated that cat and had a dramatic recovery. And that's not a clinical study, but it was a personal experience where you could see you have a fairly dramatic effect. And if you can heal the gut, you can prevent a lot of sepsis. So
Yeah.
I'm very interested in that, because horses get a lot of post-surgical sepsis from their GI tract from colic surgeries. So it's an area we could cross-study a disease that might be similar to the human.
There was a publication, a series of them, but the first one was in Nature Medicine, where they do the cecal puncture ligation model. And they showed injection of mesenchymal cells prevented mortality, something by like 50%.
Yeah, it's dramatic. I that really caught my interest to look and see, because post trauma you get lots of that kind of sepsis and problem, and I think the mechanisms are still a little bit in debate. How much of that is in effect just on boosting the immune system. I don't think these cells are directly antibacterial or antiviral, but they're clearly having an effect on septic conditions. And I think there's an interesting argument to be made that they're clearly, it could be therapeutic there.
Yeah, no, I mean, last point on that, they showed that if it was IL-10 knockout, much of the effect is gone if the cells were. But to move so quickly, I mean, I don't know about you, when I think of stem cells, I usually think of long-term chronic effects, and here we're talking about things in days, if not hours. But okay, sorry.
Yeah, so that's a good question. Can I interrupt you and make
please.
a comment there? So you could go on for 12 days with these kind of discussions, but we see in a chronic disease like arthritis an absolute immediate effect within minutes to hours to days, not months, where these cells dramatically cause the effect you're seeing on the screen right now. So if you can pull that slide up, this is a retrospective looking at canine osteoarthritis. And can you see those okay, Tom?
Yep, I can see
Yeah.
them.
So if you look at in the first 30 days, so at day 0, these dogs have a 3 out of 5 pain scale on average, and these are ones with bad osteoarthritis. Within 30 days, they drop almost in half. By 90 days, they really are in half from where they started out. So they're now down to normal to mild. Some of those literally get an effect by the time the owner gets home from the veterinary clinic. We'll get people that call us and say, is it possible that they work this quick? And I am absolutely convinced that there are 2 effects. One is the anti-inflammatory effect. Second, and it's not well described in the literature, and I'll challenge some of my colleagues if there's any of them on the show listening in, what are we doing to pain perception early on? Because I think there's a direct effect on pain that is in addition or additive to the fact you see an anti-inflammation. Just a speculation. So next year when you have me on the show, Tom, there'll probably be a publication and somebody get the Nobel Prize because they've proved what mediator that is. But it's too quick to be just anti-inflammatory. I think there's a effect on pain.
No, the pain is also a very interesting one. There was a study in the rat sciatic injury model where they were. Did you see that one? They were injecting bone marrow, and then they saw this effect. And clinically, as you know, for example, in our collaborators in Panama, they also have reported similar effects on pain. Very interestingly, the last point along those lines, there was a paper in Journal of Experimental Medicine where they were showing the innervation of the bone marrow, and that the bone marrow innervation had something to do with substance P mediated effects. Do you know anything about substance P and the stem cells?
Yeah, I do. There's actually a really good paper on that, and it's probably too much in depth, and I probably can't remember because I've got brain fade. I've read too many papers, but I've got 7 papers on pain out of the, I don't know how many 100,000 papers on stem cells, only 7 that I could find that really address the pain and pain fiber issue. And I've emailed them to a group of veterinarians that are part of the Academy of Veterinary Pain Management. And we're starting to have some really interesting discourse on whether it's mediated through substance P or one of the other 2 or 3 suggested pathways, because it could be. But I think there's definitely, and there's also potential for central pain mediation. You know, if you study pain windup, and there's, although you may have pain in your joint, there is a central mediation of that pain. And some of the drugs that we take are directly related to modulating and blocking some of those pathways. And I think these stem cells may have a role there. I could be wrong, but I just have this feeling. And the slide you're looking at right now is the change in pain medication by dogs given stem cells for arthritis. So if you look at the first circle diagram there, that's 90 days after stem cell treatment, and we tell owners not to remove their pain medication during the first 3 months after stem cell, because I don't want to change multiple things at the same time, but they do anyway. 1/3 of all dogs treated in this retrospective, this is 170 dogs looking retrospectively, 1/3 of them at 90 days after their stem cell treatment were still on their pain meds and hadn't changed. 1/3 had decreased their pain medications, but 1/3, that group in the bottom dark color, had completely discontinued their pain medication at 90 days. If you look over here in the second graph at 246 days, so basically 9 months out, still 1/3 of them were completely off of their pain meds. And these are drugs like Vioxx that cause liver damage and kidney damage and heart damage. And so this is a big deal to be able to get a human patient or an animal patient off of their pain medication or reduce the dose so you reduce the side effects from drug. So I think it's huge, and we follow this. We've got now over 1,000 animals that we've got survey data back from, and we track that, and we track it looking at young dogs and old dogs and all ages. But people ask the question. So if you use this kind of a cell therapy in a very young dog, and this is from 1 to 4 years old, 84% the owners report improvement in quality of life, and 78% of those did not need to get retreated in their first year after treatment. So this single injection lasted more than 1 year.
Unbelievable.
What about old dogs? So you got a young dog, right, Tom?
Yep.
you got a brand new little young dog. Well, and I think your colleague there, Holly, and Raj have an older dog that may have some arthritis and neurologic issues. So what about these really old dogs? Because there are reports in the human literature that when you get old, your stem cells go away. I think not true in adipose tissue. And we're finding that those cells are somehow protected from aging a little bit. So these are dogs from 9 to 18 years old. And 18 is that's even older than you are, Tom. You know, that's a really old dog. And 81% of those showed an improvement in quality of life. And these are dogs with serious chronic osteoarthritis. So interesting to look at old.
What does that translate into human age?
So it's maybe a 4 or 5 to 1. So that's an 80-year-old dog, you know, in human years. So that's an old guy. They're rare that you ever see a dog lives greater than 20 years of age. Cats live a little bit longer.
Well, obviously, the next question here is why? Why would?
Why does it work?
I mean, my personal idea here is that in the fat, you have a chronic inflammatory situation where you have different goodies such as TNF-alpha being made, leptin, and so on. And the stem cells need to somehow be protecting themselves. So I'm imagining there must be some kind of conditioning going on in the fat that doesn't go on in the bone marrow. Is it telomerase any different between a mesenchymal from the fat and mesenchymal from the bone marrow? Telomerase, OCT4, NANOG, those things have people looked at?
Yes, and I don't remember, and I can't quote the literature. It's a really good question. We should be able to answer that, and by the end of tonight, I'll find that and I'll email you, Tom, because I should be able to answer that. But the senescence is clearly different. When you take fat and bone marrow from the same animal, the bone marrow senesces faster. So one would guess that the telomerase would be different as well. But I won't. I'd venture a guess, but I won't venture data because I don't have that.
So a very naive question: do the fat stem cells, the fat mesenchymals, are they bone marrow derived?
That's an excellent question, because there is some data that shows that could be the lineage from which they originally came. And people ask the same question: well, will you inject stem cells in a normal animal, IV? Where do they go? Because they go to the lung the first 24, 48 hours and they stay in capillary beds, but then they disappear, right? They go back to where their resting point. And I think there's a little bit of data that suggests they both go to fat and bone marrow. So they go back to an ischemic area, a low oxygen content area, and just hang out. They don't continue to circulate. So they marginalize somehow and go back to storage areas, I believe.
Yeah, no, it's, I mean, one of the things in my mind always has been, you know, you can find mesenchymal cells in a lot of the major tissues of the body. And one of the questions that people have been asking on this show is, how different are those mesenchymals? Now, we do know, for example, a tooth mesenchymal has different properties than a fat, than a bone marrow. But I believe a lot of them are still originally bone marrow derived. So I guess my question, and for the clinicians who are watching this, what somebody's got to do, if you have a bone marrow transplant, a female with a male bone marrow donor, just do a liposuction and see the Y chromosome and the fat.
Yeah, I think you should be able to isolate that. I would believe that, but I think those cells have been there a long time, and they ebb and flow. So you know, as you get fatter and you have vasculature, obviously more cells come in. But interestingly, when you lose that fat tissue, so if you go on, you know, so you're gonna run and exercise, the fat becomes very concentrated, so the amount of fat goes down, but the number of stem cells per gram of fat goes up fairly dramatically. For example, a racehorse might have 10 times more than a sedentary old pasture horse per gram of fat.
Oh, wow. That's very interesting.
So keep exercising, Tom. One last study I wanted to show you, and we don't want to do too much data, but this is, you know, because I wanted to be able to show in not just anecdotal, but in a blinded, placebo-controlled study. Even though we're not regulated in this autologous business by the FDA, we did a study back in 2007, was published in Veterinary Therapeutics, Volume 8, Number 4, by Linda Black, and this was a group of orthopedic surgeons and pain management specialists that did the study with us. And what we did was we took dogs with chronic severe hip arthritis. So anybody's had a dog with hip dysplasia, this is what that is. It's bad hip arthritis. And did either an injection, it was blinded, it was triple blinded. The vet, the owner, and the dog. None of the three knew who was who. And so we truly had a blinded study. The control dogs got fat taken. The treated dogs got fat taken, isolated cells, and the treated dogs with a blinded syringe got their cells injected with a single intra-articular injection back into the hip, and the control dogs got saline, so a placebo saline injected, okay? And then we followed them for 90 days, assessing them pre and every 30 days by the owner and by the vet. So this is one of the measures. We have 5 of them. The measure looking at lameness at a trot. So when you trot your dog along going out for a walk or going to the vet clinic, this was the analysis. And the blue is the treated group. So you can see here. And the placebo is the red line. And yes, there is an absolute placebo effect in a blinded dog study. Wow. So the dog didn't know what syringe got put in, but the owner, who was excited about the study, treated the dogs differently. They spent a little more time with them. They did something. So the owner can affect how the dog feels, just like you can go home and affect how your kid feels. There is an empathic effect. So there is a placebo effect. But if you look between treated here and control here, there is a dramatic difference in lameness or pain in lameness at a trot during this 90 days. It's also maintained beyond that. This is the composite score. So if you look at lameness at a walk, lameness at a trot, pain when you flex the joint. So when you go to the doc and he flexes your joint, the range of motion you have, and your overall functional disability, again, the treated group were dramatically improved from a single injection into the joint in comparison to the control group. And last, probably really important, is safety, right? So I think you should have a whole session, a whole show just on the safety profile of cells, because there's, again, a lot of mythology out there. 3 out of 4,247 injections in the dog, including a lot of IV injections, showed an adverse event, and that was only an adverse event. Those 3 had a sore joint from injecting into the joint. They did not get systemic disease. They didn't get sick. All of them were just local joint inflammation. There were an additional 43 cases that we isolated to actually how the needle was put into the joint or twisting on the joint too much. And so, you know, now I think we've probably done 14,000 total injections in horses and dogs. No deaths, no systemic, disease in dogs and horses. And so this supports your world, Tom, in the human side. And there's a lot of data, more than probably 1,000 human cases injected without reports of any serious side effect. So it's data we share with you guys for sure.
Absolutely.
The last picture I'll leave you with, and then you can take more questions if you want. But those of you who watched the literature of working dogs, military dogs, this dog, whose name is Lex, and his handler on the left, who is now deceased, are both, he's a bomb-sniffing dog, and this guy was a corporal in the Marines, and they both ran into a roadside bomb. Unfortunately, the corporal was killed, but while he was still alive, this dog, who also was hit by all the shrapnel and was seriously injured, crawled up on top of this guy and protected him. And later on, this dog now has been adopted by the family here, and that's an honorary Purple Heart for this dog. And he was treated with stem cells about 3 months ago, and VetStem has stored a lifetime supply of cells for this dog for his service in the military. So, you know, there's so much commonality between veterinary and human both diseases and cell therapy, that it's exciting, and I really appreciate the opportunity to answer questions and try to share what we've learned in the animal world.
No, this is very, very exciting, Bob. I mean, to 99% of the population, they believe stem cell therapy is something in the future, something that we wasted a lot of money on, Proposition 71. And here you are, Bob. In total, how many? 5,000 animals you've treated?
No. More than 3,000 dogs, more than 4,000 horses. So we're probably up to 8,000 or close to 8,000 now. And these are done not at our clinic here in San Diego. VetStem doesn't even have a clinic. These are done by practicing veterinarians in everyday clinics all over the U.S. More than 4,000 veterinarians have taken our training course to be able to offer this. So anybody can walk into one of these clinics with a dog with tendon, ligament, orthopedic injuries, and other kinds of diseases, and get access to this kind of therapy. And it's only phase 1. We will have so much more learning and different other disease treatments in the future. But, you know, it works.
Yeah.
Stuff just works. And so when people get up on the podium and say, you know, so much more study is needed and we need to block it from people and, you know, go so slow, you know, being careful is important, but we were careful with our animals as well. They are, you know, maybe equally as important, at least to their owners they are. And so we watch the safety a lot, and we're very careful. And if you ever take a tour, go to vetstem.com, and you can actually take a tour through the VetStem labs, and anybody in cell therapy would look at that and say, that's you know, it's a high quality lab.
No, this is very, very interesting. One of the things that obviously comes to mind is when troops go to war, they bank their own blood, from my understanding. How far are we from having our troops bank their own fat stem cells?
Yeah, so last year, maybe it's 2 years ago now, you could help me remember, Tom, but the Department of Defense gave out $250 million to 3 investigators in the U.S. to study doing exactly what VetStem does, this adipose stem cell. And Adam Katz and the folks at Pittsburgh, and I believe at Wake Forest, all 3, those 3 groups, Virginia, Pitt, have now full programs, and they are treating Gulf War patients as we speak already in their experimental clinical trial programs to do exactly what you just suggested, which is before a new recruit would go over there, they could either get their own fat saved, or if we get FDA approval, they could use their own they could use donor stem cells to treat them on the battlefield. So what we do today could be done on the battlefield in Iraq, in Afghanistan today, treating some of these really horrible burn wounds and horrible tissue damage from explosions. And in fact, we have a program with the Department of Navy, so the Office of Naval Research, a 3-year funded program looking at wound healing in Navy working dolphins, so marine mammals. So we've already treated our first dolphin with that program and have shown that, yes, dolphins, in fact, have the same mesenchymal stem cells. And you know, they go to the Gulf and other places around the world. They're working animals just like the working dogs, like Lex. And so all of that comes together in one body of knowledge that will maybe help us with both military personnel and civilians who have similar kinds of damage from, you know, horrible traumatic car accidents and such.
Excellent, excellent. Bob, a couple of the questions here people are asking.
Yeah.
Probably the biggest one is people are going on about tumors being caused or accelerated. Now, viewers of my show will know my opinion, which is that the endometrial regenerative cell we've published, which is a type of mesenchymal cell, when you inject it into rats with a glioma is actually inhibited, which is a little counterintuitive. One of the people here put up, Bob put up a reference from a cancer research paper that says rat umbilical cord blood stem cells completely abolish mammary carcinomas with no evidence of metastasis or recurrence. So there's some evidence of stem cells inhibiting tumors. We are all familiar with some discussions theoretically why they might promote tumors. What's your opinions?
Yeah, so I've got a really strong opinion, and I have a manuscript in draft right now with several collaborators, exactly on that topic. Because as you might guess, so in the dog world, we treat a lot of old dogs, right?
Yeah.
About a quarter of all dogs that get euthanized at the end of their life die of tumors. So there's pretty high rates of tumors. So we're treating a lot of old dogs. And so we track very carefully the incidence rate of reported tumors on dogs treated with stem cell versus not. And in fact, we're at a lower rate than the published rate in the literature on tumor formation. If you look at the data, a lot of the publications showing that tumors formed in animals, in experimental animals that got stem cells, it's where they take a stem cell and a tumor line and inject them subcutaneous in an immune deficient animal. Well, I mean, that makes no sense as far as trying to then project that that's how it happens in an immune competent normal animal, because that's not how it would occur. And so we've been very interested. We're tracking it very carefully. I don't believe with these adipose cells that we use that we ever cause tumors. There is a question, if you had a bad tumor already, would it make it worse? And again, we don't see that, but we're cautiously looking at that data, and we'll be publishing hopefully before summer. We'll have that manuscript submitted and out, where we're doing this exact comparison in a very good scientific manner, trying to analyze relative risk. Is there any? And currently, I believe not. I'm not willing to go out on a limb yet and say that these mesenchymal stem cells we can use to treat tumors. There is some data that suggests that might also be true, but at least I believe, as the physician says, at least do no harm, that we don't cause a negative, and we're not promoting the use of stem cells in cancer therapy at this time. There's more work to be done there.
I mean, besides the obvious practical implications which we're talking about, it's also a very big scientific implication, which is we know these mesenchymals are angiogenic, and we know the tumor needs angiogenesis. How do we, if you have any ideas, because I have some, but nothing really crystallized, how do we reconcile this? I mean, we know you inject these in a hind limb ischemia model, the stem cells, and you get beautiful angiogenesis.
Yeah.
The tumor needs angiogenesis. Why would they not? And why would they even be doing the opposite effect that you would anticipate they'd be doing?
Yeah, I'm not an oncologist nor an immunologist, as you know, Tom, and so the science of the details of that eludes me a little bit. I think doing a study where you have naturally occurring tumors in an animal and then comparing intravenous administration of these cells, yes or no, versus controls, will help sort that out. When you're taking a grown cell line, tumor cell line, injecting it into various kinds of models, it just doesn't reflect natural disease. So if you had naturally occurring tumors, you could biopsy the tumor, you could look at tumor growth. And I think that if we had the resources and I had somebody who would supply the financial resources behind that, we could actually look at that, because there are animals with tumors who have very bad orthopedic problems who choose to use stem cell therapy anyway, and we could be evaluating that in real patients with owner consent. So it's possible to look at that, but it's a tough one.
Well, see, the great pathologist, and I forgot his name, but you probably know it, said that a tumor is a wound that never heals.
Yes.
Do you remember that saying? That the tumor is a wound that never heals. And conceptually, you would think, since stem cells accelerate healing, if you heal the wound. So my personal idea, I have several of them, but one of my ideas that I feel the most strongest about is, like you suggested, there's a very big difference between the tissue cultures, tumors, and the in vivo tumor. And the big difference there has to do, Dorvark is a person's name, D.V.O.R.A.K. In any case, the very fundamental point is this. When we discovered the tumor stem cell, by we I mean like the scientific community, a lot of data we had to throw out, a lot of the cell line data. In some ways, it made a lot of the cell line data useless because the tumor stem cell concept's giving you the notion that the cell you want to target is a non-multiplying, drug-resistant cell, which when you are throwing the chemo, you are hitting off all the progeny and all the branches, but you're not cutting the root at all. So how would you cut the root? So we've seen that in some very elegant work being performed, some companies are doing this, like Oncomed, as an example, but that a lot of the chemotherapeutic drugs don't work in the model of the tumor stem cell. And one of the main models they do is they take tissue biopsy from somebody who has colon cancer. They purify out the CD133 cells, and those ones they call the tumor-initiating cells. And when you give chemo to those, they don't do anything. But when you give things such as BMP4, bone morphogenic protein 4, bizarre combinations of vitamin D3, the retinoids, stuff like that, some of them you have to give them at concentrations that you cannot achieve pharmacologically. But the point in my mind is that the tumor stem cell can be differentiated, and that when you inject stem cells, different kinds of stem cells, they have the ability to induce this differentiation, which MediStem actually has a patent filed on, where we demonstrate very interesting data. One day we really need to publish this. When you isolate concentrated supernatant from placental cells and you treat the tumor models, you can actually see accelerated differentiation and inhibition of proliferation. And when you inject into animals, you can see inhibition of metastases when you inject the supernatant. So I was thinking it may have to do with something of inducing a healing response. But there's some of our audience here has some thoughts on this. Dr. Miller asked, have you done a dose-response analysis with any of your MSC injections to show you have maximized the positive effect of these cells for any indication?
Oh, boy, that's an exceptionally good question, and one that's the subject of a lot of debate. So when we started out on the horse looking at soft tissue, so I'm relegating this discussion now to soft tissue in the horse, we injected between 1 and 20 or 22 million cells, because we took all the cells we got from a single isolate and injected them back into every horse. So we now have a retrospective dose response study where we could actually look at, and there was no difference between 1 million and 20 million cells, and we got about a 75-78% healing rate. And in soft tissue, you get complete healing. It's not like in arthritis where there's lots of ongoing damage. You either get it repaired or you don't. So that's sort of a dose response curve. We're about to start a dose response study using allogeneic cells, so donor cells, for arthritis in the dog for our FDA submission to look at exactly that. But within the range that we use for tissue-related damage, not immunologic disease, we think we're in the range where with more cells does not give you a better response. In a disease like kidney disease, so if we're trying to inject cells IV and affect kidney disease, which we have a study going on right now at the big center in New York, I think we were too low. So we're now at a much higher dose than our initial 3 or 4 million IV. We could be as high as 25 to 50 million IV, because with intravenous administration, I think the cells hang up in a lot of other locations, including lung vascular beds. And so I think you've probably got to have a higher dose to get tissue-related cell density higher. We're also trying direct injection into the renal arteries using interventional radiology to deliver a higher concentration of cells to the kidney. So I'm probably more ignorant than I should be after doing this many animals as to dose response curves. But when we deliver the cells locally, I think we're already at maximal. But when we go intravenously for various diseases, I think we have a lot to learn still. So we're working on that. So thanks, Dr. Miller. Really good question.
Okay, there's somebody else here, Blunder from Down Under, saying that the immunology is that MSCs are attracted to the site of inflammation with high levels of chemokines, like SDF1. MSCs think they are migrating to inhibiting inflammation. In doing this, they inadvertently block immune function as they do for GVHD and in vitro T cell function. Also, inflammatory environment in vivo makes a big difference. It's a very interesting point.
Yep.
And why that's an interesting point to me is because I think, and maybe you can even say something from the clinical experiences you've had, in the case of the tumor, you have a type of inflammatory response, a systemic inflammatory response, but you do not get the resolution of that response. In the sense that, and maybe you can help me, Bob, to clarify this thinking in my mind, because you want TNF-alpha, for example. I mean, people have even done clinical trials with TNF-alpha. That the name comes from tumor necrosis factor. On the one hand, but on the other hand, it seems because the inflammation is so chronic, the inflammation is actually driving the angiogenesis. So, for example, in tumor angiogenesis, the macrophages, if you deplete animals of macrophages, either through a knocking out MCSF or other techniques, you actually see the tumor growing less because the tumor reprograms the macrophage and uses that inflammation to make more angiogenesis. On the other hand, you do want a certain type of inflammation because how else are you going to get the tumor killing? By the immune system. So there's a lot of data showing the immune system helps the tumor to grow, but also there's data showing the immune system kills the tumor. I believe that we don't really know enough about the immune system of the interaction with the tumor to make a blanket statement saying that MSCs are blanket immune suppressive, therefore they must be promoting cancer. And why I say that is there was a recent paper from a Canadian group, and for those of you who don't know, I'm originally Canadian. There was a paper from a Canadian group showing that there's type 1 and type 2 mesenchymal stem cells. In the same way that we had that Th1, Th2 paradigm in immunology and DC1 and DC2 and so on. So I think the story is very complex. But Bob, in terms of tumor inflammation, do you have any ideas? Is inflammation good for it? Is it bad? Do you think there's different types of inflammation? Any thoughts on that matter?
Yeah, there's clearly different types of inflammation, and since we don't work in tumor therapy, I probably can't tell you as much about that. But if you can you call this slide up on the screen, Tom, where you can show that this is a picture of a tendon.
This picture of the tendon, that one, yeah.
Okay, so this was a blinded controlled study and gets to the little bit to answer this question. So if you look at, this is an injury in a tendon where you have very severe, 50% of the cross-sectional area of a tendon in a horse is damaged, and we inject either saline control or stem cells. And the top, that's the saline control. And I know most of our audience probably are not histologists, but all the little purple dots there are all inflammatory cell infiltrate. Very dramatic in the saline control, and under polarized light to the right shows the scarring, those lines. You can see the scarring in there.
Wow.
Unlike if you look in the stem cell treated group down below, the pink lines there are normal collagen, and although there are a few little purple dots, the amount of infiltrate, cellular infiltrates, dramatically reduced. And Artie Kaplan was one of the ones who looked at the slide with me and said, you've reduced the cellular infiltrate, you've reduced the number of white cells in there that are dumping all their lysozymes and other damaging enzymes, and so you're protecting tissue and growing new tissue, probably both. So somehow the cells know how much inflammation to downregulate. When you put them in there, they don't turn it off. It doesn't go to zero because there is a natural healing response, and the macrophages are in there cleaning up damaged tissue. So I can't probably be as smart as I should be about what the cells are doing, but clearly they are looking at and regulating the inflammation appropriately, and they didn't immunosuppress that tissue. They just downregulated an overactive tissue. And maybe in tumors you're getting the same thing. Clearly you have the ability to produce new blood vessels and be angiogenic, but at the same time these cells are causing tumor cells to differentiate and slow down growth. So they're trying to manage what is a disease condition when you put them in there. And I think that may be where your positive's coming from. But the amount of inflammation in tumor and the interaction there, I don't know. But I do know that we do not see an increase in tumor level in dogs given intravenous and intraarticular stem cells. And these are dogs that are older that would have a high propensity to tumor.
Mm-hmm.
And in their lifetime, a number of them will die later. If the tumors were getting impacted, where we were growing more tumor, causing it to accelerate one that we didn't know was there,
Mm-hmm.
we would have certainly be able to measure an increased rate, and we don't. We measure a decreased rate. And so, that's just observational in our population of old dogs that get a lot of tumors. But, and it doesn't speak to the cellular level, but it speaks to the clinical level.
Now, at the clinical level, when you treat, let's say, some of the inflammatory conditions that you treat, you mentioned the polyarthritis before we went live here. Let's show to the audience that nice little graph, actually, that data, which I thought was very interesting.
Oh, there we go.
There we go. Let me pull it up so I can see it. So over here you have 21 animals that were chronic and treatment refractory. So as you said as you told me before, these ones had steroids, cyclophosphamide, some of them had, you said?
Cyclosporin, which is atopic, which is an immune suppressant. And they had lots of pain medications as well. So these were, they're not really quite the same as rheumatoid arthritis, but it is an immune-mediated polyarthritis, so multi-joint immune-mediated arthritis similar to rheumatoid arthritis. And all of these were treatment refractory, meaning the treatment just wasn't working. And so we treated them with intraarticular and IV, all of them IV and part of them intraarticular as well. And we got 90% that had an improvement. 57% or 12 of these 21 had a significant improvement. So these are ones that the owner and the vet reported as significant improvement as opposed to mild. So really only of the 21, only 2 had no response or got worse from their immune-mediated arthritis. And these are ones that were not being treated well and had a lot of pain. And so it's just an interesting, and they have a lot of inflammatory disease going on in the joint. So as you know, in these, it's just like in humans where they have horribly inflamed and painful joints. And this therapy worked surprisingly well. And that's why we sort of shared that data with you and your group and published together in your rheumatoid arthritis paper. But it is a similar kind of disease and highly inflammatory.
Are you going to publish this, or do you want to do this one together?
We have the data now ready to go into draft, and it would be a great honor to publish this with your data. At the last time when we published with you on rheumatoid arthritis, we didn't have as much of this data, so it would be a great one to put out together, because these are real walk in the door. These are not mice in laboratories. These are walk in the door patients with all the other problems that patients have. And our patients just happen to have 4 legs, so they have 2 more legs to get sore than you guys do.
Yeah. Well, this is, my question where I was going with all of this before I ask you the next question is, with this, different systemic inflammatory levels, SED rate, CRP, things like that, do you see decreases? Have you looked at some of the systemic markers?
So in these, we have the standard inflammatory markers, looking at fibrinogen and looking at C-reactive protein, and they will go down. Joint fluid is very indicative because these have high protein levels, high cellular, mononuclear cellular infiltrates, and so those will go down as well. And just the overall swelling in the joint goes down as well as just the cellular infiltrates.
Okay, now let's go back to a more philosophical question. This is some pretty convincing data, and the safety profile is established. What do you think is stopping us as a society from starting to use this in the U.S. as an autologous treatment? I mean, we see the group at Cleveland getting a couple $1 million grants to do autologous bone marrow expanded MSCs for multiple sclerosis. This seems like such a simple procedure. And you already mastered the procedure in the large animals and have the data. What is the next step to, let's say somebody who's watching this, who's an extremely wealthy person and who wants to accelerate this, what would be the next step to getting these things done in the U.S.?
Well, I mean, the companies are following those steps, and the FDA, I think, unfortunately, has decided that even autologous cell therapy in the U.S. is somehow to be treated like a drug.
Uh-huh.
And yet today, Tom, you could go get a kidney transplant, right?
Yeah.
You could go get a liver transplant. There is absolutely no FDA regulation of tissue transplants, and those tissues are full of stem cells. But when we gave it the name of stem cell, because of the background, people thinking about embryonic stem cells and donor stem cells,
Yeah.
somehow we got autologous cell therapy lumped into into the category of all the rest of cell therapy and being treated like drugs. I think it's near on to criminal,
Mm-hmm.
because you should be able to use your own stem cells just like you could get a skin transplant or a kidney transplant. You know, moving your own tissues around. It's done in plastic surgery every day, right?
Mm-hmm.
You know, millions of people have had their fat and even enhanced fat transplants done. Yet, because we called it stem cell, and there was some other isolation technique, and you know, it's not I'm suggesting anybody should be able to do this. It needs control, but I think it clearly needs a different pathway to allow more rapid access to your own cells. I mean, you're just moving your own cells around and using them, in particular in wound healing, you know, diabetic wound healing. Oh, God, it makes so much sense to be able to use your own tissues to be able to heal yourself. And,
Oh,
you
yeah, but
know,
you're talking about non-expanded, right? Or do you think
yeah,
expanded as well?
non-expanded for sure,
Yeah.
and even expanded cells, if they're done in a controlled environment. Yeah, I just think the pathway should be easier to get to the market and get to the clinic than treating it like a foreign drug that's a synthesized drug. You know, they're not the same. They
Yeah.
really are not the same. You know, the more you do something like growing cells, the more you've got the chance you do something to them. But, you know, if this is not donor cells, there's a lot of issues that are now removed, in terms of issues and safety issues. And I just think we should maybe be a little bit more lenient for self therapy, particularly for serious kinds of diseases. You know, drugs that are highly toxic are fast tracked for cancer patients.
Mm-hmm.
Would make sense that for certain diseases, for example, ALS, and you've been involved, Tom, in looking at Lou Gehrig's disease,
Yeah.
you know, being able to get into therapy sooner with those, you know, chronic serious MS, chronic serious other diseases that are immune mediated, where the patient's going to die, using stem cell therapy, particularly autologous stem cell therapy, I think has huge opportunity and could allow us, based on a lot of the data that we're showing in veterinary, that we should maybe fast track for those people, because there are people going offshore every day to get this therapy. And you know, you're well aware of that. They're going to Panama, they're going to Australia, they're going to Korea, they're going to China, they're going to Japan. You know, not all third world countries that
Yeah.
are fast tracking autologous cell therapy, so that you could use your own cells to try to help heal yourself.
No, exactly. I agree.
Opinion.
What, my opinion? Yeah, my
No,
no,
I'm
my.
just saying,
Oh,
opinion only. And so,
oh,
yeah,
yeah, yeah,
you know, you've been involved in this a lot.
yeah. No, no. My opinion is definitely there should be some regulation, especially when you're expanding the cells. But with autologous fresh non-expanded, I agree with you 100%.
Yeah.
Okay, let's see here. I think we covered most of them. And one last question, I guess, for me, and then if nobody else has any questions, we can wrap it up. Um, Bob, what about cachexia and conditions associated with cachexia? As you know, my mom passed away from cancer in September, and it really struck me as something very crazy that what she died from was actually the
She
cachexia.
starved. She starved to death. Yeah,
Still,
and many in old age, most of us starve to death in the end. It's true.
yeah.
Yeah.
And I'm thinking along the lines of two areas. Um, One is nothing seemed to have worked for cachexia, period. Number two, cachexia seems
Yeah.
to be mediated highly by this chronic inflammatory state, TNF-alpha and so on. But number three, you've shown this nice data with the muscle generation. Obviously, the big thing that's happening in cachexia is muscle loss. Have
Yep.
you seen in some of the degenerative conditions that you've treated that might be associated, because it's not just cancer cachexia, right? You get cachexia with COPD, I think, as well, and some
Yep.
other conditions. Any chronic inflammatory cachectic conditions that you've seen some effects, or any thoughts on this?
Yeah, so be careful how you interpret what I'm going to say, okay, Tom?
Okay.
Because and everybody that's listening, because the cachexia you see from various kinds of chronic diseases may even have different mechanisms, right? So I'm going to just quote observational data that when we take animals that are in significant pain, animals that have significant chronic inflammatory disease of various kinds, and they're treated with stem cells both locally and intravenously, systemically, we see several things happen. One is you clearly get a reduction in pain. Two, you get an increase in activity and energy level. In study after study, we measure activity in a lot of different ways. So, activity, quality of life, happiness of the animal, playfulness, the things that they do that we measure that are very real, that actually can be quantitated by the owners in some of these studies. And so they clearly feel better. They also will get better muscle mass and better hair coat. So those of you who can see me on the screen can see I probably need stem cells on my head for my hair coat. But there is an increase and reduction in inflammation in the dermis. So you get better, you get healing of microvascular disease, I believe. But you also get this increase in muscle mass, increase in appetite. Now, does that come from just a reduction in pain? It's possible. So if you are in pain, you don't want to eat, you don't feel like exercising, you don't build muscle mass. If you reduce the pain and inflammation, all those things may be the sequelae and come after that. But there could be an effect on appetite or on muscle mass that is independent of those mechanisms. And boy, I can't at all speak to science behind that. It's just observationally you see animals gain weight, gain muscle mass, in particular in painful joints where they've got a reduction in muscle mass due to lack of use atrophy.
Hmm.
And so, is there an effect on cachexia? And those mechanisms can't speak to the science, but observationally, we get an improvement in those outcomes. And if I was cachexic, I'd be treating myself with stem cells, even if you just feel better. Maybe you move around, which builds muscle mass, which increases appetite, which increases muscle mass, and it all relates to each other. Highly speculative, I know, but I'm not suggesting that stem cells have a direct mechanism that would reduce cachexia by some specific mechanism. I just don't know that.
No, this is a very, very important conversation, Bob, that you and I should continue. We should continue offline. One last parting thought.
Yeah.
It just occurred to me right now about this business with the stem cells causing cancer. The first use of mesenchymal stem cells was in cancer patients. Do you remember
Yeah.
that? The original studies with I forgot who it was. They were giving breast cancer patients who had a lot of chemo. The original publication. I think Kaplan was one of the authors of it.
Yeah, and if you think about what are one of the things that these stem cells, mesenchymal stem cells, clearly do, they help support bone marrow hematopoietic production, right?
Right.
I mean, there's 100 studies that show that bone marrow transplant patients, the implantation or transplantation take rate, the engraftment rates go up when you co-administer with MSCs. And you can look at it, and just even in the bone marrow. So an MSC and an HSC
Mm-hmm.
sit on an HA molecule, right?
Yeah.
Hyaluronic acid molecule, and they're supportive. They're supportive in cell culture. They're supportive in the bone marrow. So what's one of the biggest problems that we cause by chemotherapy in a cancer patient? We
Ex-
absolutely wreck their immune system. So if you think maybe these cells have a role in supporting the immune system while we're trying to kill off cancer cells, which are, I guess you could inherently say they're part of the immune system gone haywire. But, how many patients die from pneumocystis or 10 other, kinds of infections that normally you and I would never get because we wrecked their immune system. And, it's a race to kill the cancer before we kill the patient. Maybe there's a role there because these cells clearly support immune system function. So in AIDS, in viral diseases, in bacterial diseases, could we use them in a manner that would help support immune system function? I don't think there's enough research in that area, but I think there's certainly a possibility. And part of this show, I know, Tom, you're trying to generate some controversy and speculation about how the cell therapy industry might go forward, and that's one of the areas I think could be very interesting.
Well, the last question, I keep saying last question, is for hematopoietic transplantation. To me, it would seem very simple to do autologous adipose therapy, especially but clinically speaking, for GVHD, would these patients have enough fat? They would, right?
They would, because you don't need a lot. Now, if you're trying to do it autologous fresh stromal vascular fraction, you need to get enough to get a dose out of there too. And that's not a lot. I mean, there's, again, we're giving a dose in a big dog of 3-4 million cells, 5 million cells, and there's 500,000 cells per gram of fat. So even 10 grams, which is the size of your thumb, the tip of your thumb, is enough, probably to get enough cells out for that dose. So for hematopoietic transplantation, and as you know, Tom, because you wrote the paper that I gave for you at the veterinary surgeons meeting on solid organ transplant, using these cells to reduce the rejection phenomena and support engraftment of a new organ is a big area of research and clinical endeavor right now to see whether that might work. So both hematopoietic bone marrow transplantation and solid organ transplantation both potentially can utilize the role of this.
Excellent. Okay, Bob, any last things you want to say before we wrap it up?
Boy, other than I really applaud your effort to continue to get discussions and bring these topics to the surface, because even though somebody like Arnie Kaplan would tell you, although he's been around it for 20, 25 years, I mean, in 1982, he was telling us we would find these cells, right? So we're going on 30 years. He also would tell you how little we know and how valuable. I mean, he's been encouraging me for a decade now, how important the veterinary data is to help us at least create good questions and hypotheses that we then can test and go back and maybe bring some of these things to human clinical trials and help patients, because in the end, that's what this is all about. And it's exciting for me to see what's happening on the human side, and every disease you could name, there's data.
Exactly. Well, thank you very much, Dr. Harman, and I want to thank everybody for watching, and thank you, Raj, and the people at The Latest With for setting up this format. And we'll see you again next Tuesday at the same time and same place, 3:30 on Tuesday. Thank you very much. Bye-bye.